In the last 15 to 20 years, there's been just incredible DNA discoveries, and as soon as they sequenced the human genome, then they did chimps and then orangutans and gorillas, and then what do you do if you have genomes? You start comparing them, right? So I've been debating creationists for 40 years. I have a graduate degree in biology, in ecological entomology, and then I went to medical school, and then all this DNA stuff came out, and then I re-engaged. And so everything I'm going to show you, I've vetted and been debating on social media, and some of the best apologists I could find. And this stuff is really holding up. For the first time in 40 years, they do not have an answer for these. They think they do. So we're going to go through, if I can figure out, and by the way, I usually do it from my laptop on Keynote, and then this is converted over to Microsoft, so I don't know how well it's going to do. Let's find out. Okay, so this is going to get a little technical this morning, but I put it all on a website that I just started about 6 months ago. This one here is called truthfulorigins.info, and so there's just everything else we're going to talk about is on there. So if this goes a little too fast for you, or you get bogged down in the jargon, you can go to this one. And then I've also been teaching some of this stuff at the public school level. And so I took all of my religious stuff, why Christianity is not true. There's 4 reasons. There's errors, contradictions, adulterations, and Yahweh is an immoral, innocent child killer. And so if you're going to refer to the Bible as a source of morality, it's horrible. So anyway, that's all on this one, which is that info up there. And then if you have anybody who you encounter that is having trouble with evolution from their indoctrination from religion and stuff, this one is just the science. It doesn't have anything on religion in there. I've scrubbed it as much as I can so it's more palatable for a lot of people. So I'll show you these at the end too. Let's see if I can keep going. Okay, so before you even start a discussion about origins and species origin, some things I usually stop and I take a breath and I say, okay, who's asking? Is this an apologist who's going to go into this debate with you in a totally different agenda than you are? You are probably just wanting to find out what's true or how does this argument hold up. They're going to use you to find out what the latest science is so they can go find out where you're wrong. So if you get into a debate with an apologist, just realize that you're in 2 different agendas, and his or her agenda is not going to be the same as yours. So if you're on a one-to-one debate with an apologist, it's probably not going to go anywhere. They're going to be parasitic to you. They're going to suck you for all this new knowledge. And so, but if you got somebody who has honest curiosity, great. The other thing is, what type of creationist? The last thing you want to do is get involved in a creation debate and find out that you're arguing against a young Earth creationist you thought, and this guy is Reasons to Believe with Hugh Ross. He's an old Earth creationist, and you just wasted 15 minutes of your time assuming things that aren't true. So there's all kinds of different creationists out there, and people will merge their worldviews and their creationism together into this mosaic thing. And so just like Matt was saying, you got to stop and say, okay, what are your beliefs and what do you believe? You know, take time on that. Are they more interested in intelligent design? There are people who are not religious who say, what's wrong with intelligent design? I've written a nice blog on that. It's only 15 or 20 minutes that reading. Who's asking? Is it a family friend or an apologist? Do you really want to poison a relationship possibly with your family or a close friend by going down this road, by opening this door? Because people will take their belief systems and the ones that are really foundational for them, and they'll merge them with their identities. They'll become core beliefs. They'll elevate them to sacred beliefs. So when you start attacking where humans come from, and they start realizing that that's the brain feels like it's an attack. And so do you really want to be attacking family members or somebody on their deathbed who's going to, die, and Majima says, well, what's going on here? And do you really want to at that time have a discussion with her that actually, well, you're going to be gone and there's no afterlife? So those are things that you may want to talk about. One one area that I do kind of go after them is if they're in a leadership position. If they're affecting younger people and leading them down with lies and going the wrong directions, I kind of tend to go after those people a little bit more. Seminary professors, stuff like that. And so then I'll be a little bit more aggressive, but those are questions you have to ask. So the newer discoveries, in my mind, are these four: shared endogenous retroviruses, human chromosome 2 fusion, shared DNA repairs that I just discovered a little while ago. This book is unbelievable. This is written by a theist. He's a cancer researcher in the New Zealand area. This guy has done an amazing job showing why DNA shows that we're related to all the other great apes. But he doesn't have much on human chromosome 2 fusion, but the other stuff he has is great. And then the last one is shared pseudogenes. And these are all on that website I told you. You can go there and look, and their graphs, everything is cited, so you can go check the original sources and stuff like that. Except the shared pseudogenes, I don't have that yet. And the reason why it's not up yet is that my site is really a contra-apologist site. So if you are in a debate or you're going into someone or you say, I don't know the answer to that, I don't know why that gene that you're talking about in the fusion point, how that's addressed, it's in my site. I've taken every single creationist objection I can find and found the answers to them. And it's not easy. It's taken years sometimes to find that. So I don't have the shared pseudogene up yet because I haven't researched all the various creationist supposedly comebacks for it. But it will be coming up. And my site, there's no clickbaits. I don't earn any money from it. So I'm not telling you about it just because it's going to be an advantage to me. The other thing I should tell you is that if for the people who don't want to get into some of the weeds about DNA, also, the other thing is whale evolution. The evidence for whale evolution is unbelievable, and it's visual. I mean, you actually can see pictures of the fossils and stuff like that. So the other thing I did was I made a set of 3 videos. They're 26 minutes long. The first one is living whales, the second one is fossils, and the third one is DNA evidence again. And so, unfortunately, I'm not the dynamic presenter I was when I was in my 20s, but if you're heavily caffeinated, you can probably get through those 3. And the reason why I did a video instead of all this other stuff I'm doing is because it's better to show the whale evidence rather than just talk about it because it's so powerful. So if you have somebody who doesn't understand what's this stuff about evolution, what's the evidence for it, why do hundreds of thousands of scientists all around the world accept it? You can say, well, I got this crazy guy who did these 3 videos, and there is stuff in there that you will not find anywhere else because I spent the time to research it and dig it out. And so that's the other one too that you want. Okay, so real briefly, you guys are college students, so hopefully this is a little bit easy, but remember that if you take chromosomes, when they're about to form a daughter cell, the DNA all contracts down into these compact luggage pieces that can be moved around. There are some ropes that attach, microtubules that pull these apart. But if we start to untwine the chromosomes, they'll form this structure which has a loop onto itself. And eventually you get to these protein balls, and there's a couple of loops around those. And if you keep unwinding and unwinding and unwinding, you eventually get to the structure that we all know as DNA. And it has 2 backbones, and then there are 4 bases, right? ATCG, A bonds with T, C bonds with G due to the hydrogen bonds. And that's basically DNA. And everything about life and running all the structures of biochemistry of life comes from those 4 bases. And there's 3.1 billion of them, which is why it's so complex. Okay, so I'll just give you a sample of what we're not going to talk about. This is human chromosome 2. And if you take the H stands for human, the C stands for chimp. If you take the chimps and the human chromosomes and line them up, the first thing you start noticing is that they have very similar banding. And so there's something going on here that the genes are all lining up. except that all the great apes have 48 chromosomes. We have 46. So the question in science for a long time is, how do you go from, if evolution's true, how do you go from 48 to 46? You can't lose that much DNA. The organism isn't going to survive. So basically, it was not lost, it just got rearranged. It's like you're going on a trip and you have, instead of 3 bags, you got 2. And so what happened was the ancestor of humans, and they had 48 chromosomes also. We know that from the fossil record. And what you do is, if you take chimp chromosome 12 and stick it onto the head of the 13, you basically get human chromosome 2. So what happened was there was a fusion between not chimps, but the ancestor of chimps and humans. The 12 and 13 fused together to form 2. All the DNA's there. And this is actually pretty common. It occurred through something called a Robinsonian translocation about 1 to 3 million years ago. And this is something that actually occurs naturally. So it's about 1 in 1,000 people have 45 chromosomes instead of 46. So where I'm from, Portland, Oregon, it's about 2 million people. There's 2,000 people walking around with 45 chromosomes instead of 46. They're normal. They have all the DNA. It's just, the problem they're going to have, if they don't know it, is what's going to happen when they try and have children. You're going to have somebody with 46 mating with someone with 45. And that's a 75% chance that there'll be something wrong, like Down syndrome, or it'll die. And so on my website, this goes into a lot of detail, how it was found. Here's the tips of the chromosomes have telomeres on them. And so when they saw this, that the banding lines up if you stuck 12 and 13 together, then they made some predictions. And the predictions are that in the middle, my pointer doesn't work either, in the middle you should find the tips in the middle instead of on the ends. And then also this centromere here is the one that's working, but there should be a second one that's been turned off. So there were predictions that came as soon as they found out what's going on. And indeed, that's exactly what they found. And when you sequence this fusion point, you get the telomeres here. They have a specific TTA, GGG, and then the complementary on the other side. And you can actually see this is the fusion point right there. So we are looking at something that happened a couple million years ago, and we actually get to see it. This is very powerful. And the reason I don't use it, it's not my go-to, is because if you're a creationist, you can just say, look, the chimps and the gorillas were made with 48, and then when the creator or the designer was making humans, he just rearranged it differently. It doesn't really tell you. We need something more. We need something that is so solid that they can't wiggle out of it. And that's why we're going to endogenous retroviruses, because as far as I can tell, they're not getting out of this, but they're trying hard. Okay, so endogenous retrovirus stands for endogenous, means it is established throughout the entire population. And a retrovirus is a virus that, instead of going from DNA to RNA, it starts as RNA and it goes backwards. Because normally you have DNA, you make messenger RNA, it goes to the ribosomes, and you make proteins. But this virus, in order to insert its DNA into your DNA as a host, it has to reverse. And I'll show you, I'm going to give you the terms here so we don't get lost in the labels. But in a minute, if it works, I think it's going to work, I'm going to show you a 5-minute video by Howard Hughes Medical Institute, which is a life cycle of the HIV, the virus that causes AIDS. And all the retroviruses have the same fairly simple genetic code. They have 3 genes, GAG, POL, and ENV. Sometimes they have a 4th one that people list, but it's basically 3 genes. And then it's flanked by something called LTRs, and this will become important in a minute in the debate with the creationists. So when a retrovirus wants to infect you, it gets into your through your defenses, and it binds to the outside of your cell, gets absorbed inside, and then it converts its RNA to DNA, and it picks a site to cut your DNA, pull it apart, and then splice its DNA in. Okay, it's like you cut a rope, and then you've got two. When you get it done, you've got two knots, right, on either side. If you're putting a piece of rope between two other ones, and so these sections here are called target site duplications. You can find these in your genome. There's 440,000 of these. Your genes are, your genome is made up of 8% ERVs. Throughout millions of years, we have been bombarded. How many people get virus attacks during the year? You know, so it's a lot. So these are specialized viruses. These viruses insert their DNA into your DNA. And once it's in there, you do not have the mechanisms to get it out. So this is an example of an HRV, human endogenous retrovirus, and it's in the K10 family. And it finds a target site that it wants to insert. And there are some host preferences. It likes chromosome 19, and they like to insert in lines. But the insertion point is random, and it's localized. It's like an address in a city. Just because you're in the same city doesn't mean that you all live in the same house. So this thing inserts its DNA after it makes it from RNA, and it inserts it randomly. And then once it's there, it's there. Now, these LTRs, it has to make them, and we'll talk about this in a minute. These are promoters, and in order to promote the reproduction of this virus, it usually it doesn't have promoters. If it just inserted just the genes, it wouldn't go anywhere because it's going to use the host enzyme, and so it has to, in order to reproduce itself over and over, it has to have promoters, and it has to make them. We're going to talk about that in a minute, because this becomes really important in the debates. And then there are 3 genes, and the GAG make the capsid like a lunar module. The pol are the enzymes that it uses to cut into your DNA and to also to make the DNA from its RNA. And these 3, and then the ENV are the envelope proteins that are made. So it's actually a simple, in some ways, parasite that it's going to get in there. But these are the terms that you know. So you know that it's cut in because there are 2 target site duplications on either side. And the LTRs come from the virus. They are not original. And this is the crux of the argument from the creationists. And I think, let's see. I don't know if this is going to work or not. If it doesn't, I will. Nope. Okay. So I have, let's see how you guys, where did it go? Oh, here it is. See if I can make this. I'm a Mac guy, so it's like, okay. So this is less than 5 minutes, okay? You just want to get the overall thing, and you want to kind of get the LTRs and the fact that it's cutting in with an enzyme. Okay? Reverse transcriptase is going from RNA to DNA. Integrase is cutting the host DNA and splicing it in. So let's see. The other thing I don't know about is volume. Let's see what happens.
So this is HIV. It's a typical retrovirus, meaning that it has an outer envelope. And in the center, it has 2 copies of RNA, as well as an enzyme here in blue that's reverse transcriptase, which will ultimately turn that RNA into DNA. The virus itself, with this outer envelope protein, actually directly infects T helper cells. The way that it does this is that as it comes up to the cell surface, it uses receptors that are on T helper cells and exclusive to T helper cells, which are CD4 molecule, which really defines T helper cells. It's a surface receptor that binds to the envelope protein. That causes a conformational change and allows a second receptor to grab hold of the envelope. This is the chemokine co-receptor. It's also called CTR5, and we'll talk about that more. What happens now is that the stalk of the envelope protein pierces through from the virus into the virus has a matrix and a capsid protein shown here in green and red that essentially are digested when it enters into the cell. That releases the viral enzymes and the viral RNA. And here we have reverse transcriptase, which takes the viral RNA and using host nucleotides converts that viral RNA into a single strand of DNA. While it does that, it makes some random errors, which is characteristic of reverse transcriptase. It has very poor proofreading activity. That single-stranded DNA now is again reverse transcribed into a double-stranded DNA. At that point, another enzyme that has come in with the virus in the beginning, called integrase, essentially grabs hold of that double-stranded DNA and carries it through a nuclear pore into the nucleus of the cell. Within the nucleus of the cell, it finds the host chromosome, and it basically, the integrase enzyme, makes a nick in the host DNA and allows for HIV to insert itself into the host chromosome. And that right there is what establishes lifelong infection. Now, RNA polymerase comes along and makes messenger RNA. Those messenger RNAs encode for different viral proteins. They end up associated with ribosomes at the surface of the rough endoplasmic reticulum. And here's a piece of mRNA that's making envelope protein, which is directly produced into the endoplasmic reticulum. And it's shuttled them through the endoplasmic reticulum and taken to the cell surface, where at the cell surface it becomes embedded in the cellular membrane, and at this point, coalescing with other envelope proteins that have been produced, you have this cluster of envelope proteins now on the surface of this infected cell. At the same time, there are other messenger RNAs that are being produced that allow for translation of other viral proteins. So here are additional viral proteins being made, which are going to be used to make up the key components that the virus ultimately is going to need. These are transported again to the cell surface, to the area where these envelope proteins are, and a strand of RNA as well as some of the enzymes are part of that complex. This then buds off at the cell surface at this point, but it's still not a mature virion because the polyprotein chain needs to still be digested into its component parts. That's done by an enzyme called protease. Protease breaks up those polyprotein chains and ultimately allows for them to coalesce and form the mature structures that make up the final virion. And now you have a mature infectious virion that can go on now to infect other cells. Once that happens now, the cell can produce tons of viruses, and this is really what then keeps the whole process going.
Okay, hang with me. We're going to summarize too here. Let's see if we go back. Let's see. Okay, so remember that in cell biochemistry, you're trying to make proteins, especially enzymes, because they control everything in the cell. And proteins are made up of amino acids, and amino acids are assembled like a bunch of railroad cars at the ribosomes, and the ribosomes are getting their messages from the messenger RNA, and the messenger RNA is just a copy of the DNA. So this is the enzyme that is being used to make messenger RNA. It's a host enzyme. It's not an enzyme that the virus is using. And its job is to read the sequence here on the DNA to make amino acids. It doesn't really care about making promoters or anything, or regulatory elements. It's just going to make the amino acids. That's all it's reading. So if the virus did not make LTRs when it infected it, it wouldn't be able to go on and make further infections. That's where the LTRs come in. There are a whole bunch of promoters that the virus has to have there, or it couldn't make infections that would go on. This is very, very important because it turns out of the 440,000 ERVs that we have, 90% of them are LTRs. The LTRs like to swing around and merge with the other one on the other side and make solo LTRs. And then the rest of the virus that have been there for millions of years kind of goes away. So the creationists are going to argue that these LTRs are native, that they were put there by the great designer. And the reason why chimps have 200,000 LTRs that are in the same exact place as we do is because that's part of the great design. But the LTRs are only inserted by the virus. They're going to try and argue back the other way. Okay, so this is a busy slide, but I'm just trying to show why the LTRs are coming into your body. You already have all the promoters you need. It's already built into your DNA. These are parasitic promoters that are there for the virus. But our body is not stupid, and natural selection will use whatever it needs. And promoters are great. They can kind of keep the DNA going. And so what has happened is that through millions of years, the host, us, have taken over these LTRs and we're using them. This is how we use to get a lot of things done. And the ENV gene that makes placentas, the interface between the fetus and the woman, is derived from an ENV gene. There's also a very famous gene called an ARC gene that is important in our brains in terms of signaling, and that comes from a GAG protein that came from these ERVs. So your body is not beneath itself to use enemy parts for its own use. And so that's really what's going on. Not that the LTRs were there by the great designer for the beginning. It all comes down to an argument between common design and common descent. Okay? All right, so here we go. This is like the summary slide. This is not, the jargon, the biological jargon is easy to get into it, but it's actually a very simple concept. Okay? We have found that 8% of our genome are contained with ERVs. Most of them are LTRs now after millions of years, like 90% of them. We know that the retroviruses insert randomly by location because we have retroviruses right now that are infecting people and koala bears. And if you do Wikipedia, there's lots of different retroviruses. So we know that they insert randomly. They pick out different parts. Now, they do have preferences. I think chromosome 19, and they love to insert into something called a line. But overall, even within those, they're very specific about where they insert. They need promoters, which is why they make LTRs before they insert their genes. We have identified 8%. 200,000 of them are shared exactly with chimps. Same location. Some have the same mutations. You know that when you compare chimps and humans, that there are 200,000 of these that are identical. Now, if you're inserting, if you're shooting at a target or you're shooting arrows at something, and you find out that there's two targets that have the exact same holes, what's the mathematical possibility of that? It's not mathematically possible. The only explanation is that they had the same origin. Somebody copied one of the targets. Or if you're a teacher or a student and you hand in a report and it has the exact same paragraphs as sentences, what are the odds? Maybe it was an online course and somebody was in Massachusetts and the other person was in Pakistan, and you can prove that they didn't communicate with another. If you have the same report with the same sentences, they had to have copied it off the internet, off of an original. Somebody else submitted or got it. So the ERVs are in the exact same locations between chimps and humans. You have to explain that if they're randomly inserted. And when they insert into the host genome, you have those cut ends, those target site duplications, which I'm going to actually show you one in a minute. And these TSDs are on either side. So the creationists who are arguing that all these LTRs are original, they were not put in by retroviruses, they have to explain integrase, and the reverse transcriptase, they also have to explain these 2 target site duplications on either side. And that's where they really start to rationalize. So this is it. If you find 200,000 ERVs in these same locations between 2 different species, the only rational explanation, since they're inserting randomly, is that they had to have inserted before the species split. That's the only rational explanation. This is what's been holding up in my debates with them. They'll try and say that the LTRs are original. Okay, so, and then we already went over this one with the plagiarism example. Okay, so here's one here. This is an endogenous retrovirus. It's in the K105 family on the top, and the one is a different one on the bottom. Can you see? This is the target site duplication, target site location. And then can you see how it got duplicated? Here's the ERV. This is the exact same ERV found in humans, chimps, and bonobos. Same ERV, target site duplications. You know exactly where it hit because the duplications are on the side. This one down here, again, here is the target site, TGA, GA. Here it is here, duplicated. Here's the ERV between these. The orangutan doesn't have it. What does that tell you? If these LTRs are supposedly designed, why do some species have them and some species not? So this tells you that this ERV inserted after these other apes broke off from the orangutan. It's the only explanation that fits rationally. Now, if you take a whole bunch of ERVs, and remember there's 200,000 of them shared by chimps and humans, the same ones. There's 440,000 of them. And you look at these, here's some here in different forms. These are all common to humans, chimps, bonobos, and gorillas. So is this group. These are common to everyone, but these here are not found in orangutans. These are found in all of these groups, but not in gibbons. These ERVs are found in all of them except New World monkeys. So you've got thousands of these ERVs. They're sequencing them. They're starting to compare them. And all of a sudden, you start to notice when you start to put which ones are present in some and which ones are not, you end up getting a phylogenetic tree. There are phylogenetic trees from fossils, from ERVs, from DNA scars, from pseudogenes. When you get phylogenetic trees from different independent sources of science and they're all saying the same thing, you've basically got evolution to the point of proof. Not only that, but if you're debating a theist, an Abrahamic religious theist, eventually, and a lot of stuff what Matt was saying, you know, I felt like raising my hand. No, I have some answers for you. Eventually, they will always go back to one thing, the fall. How do you explain all the disease, the cancer, and everything else we have in the world? It's because of the fall. Adam and Eve fell, and then that introduced all these problems that we're seeing. Genetically, all the genetic diseases are like this. So there's 2 ways that science disproves that. Number one is that we know from genomics that the human population never went below 3 to 10,000 in Africa. They have the genetic data on that. So there never was a primal couple. 2 at the beginning, and there never was 8 off a boat. You can't go back that far. The other thing that happens is that if you're going to say that disease entered the world through the fall, you're not going to be able to get this. Because all those diseases, the cancers, the infections, all the things that are wrong with the world would be like a scatter plot. You know, it would just be all over the place. But this is a group of parasitic viruses that are trying to take over your DNA and kill yourself. You know, that's what happens in AIDS, right? Without modern medicine, it's terrible. That video that you just watched, those CD4 cells are immune cells. Unfortunately, that virus happens to hit the immune cells. And then there's a race between the virus making progeny and killing you, and then your immune system trying to control it. And after 8 to 10 years without intervention, people with AIDS die. So you've got this is called a nested hierarchy. You've got a phylogenetic tree, and the fall would not produce that. So I assert that science has now disproven Adam and Eve historically, and it's also proven that the fall doesn't fit either, because we can make phylogenetic trees. So if there's no Adam and Eve to fall, there's no original sin, all of Christianity and the Abrahamic religions just fall flat. This is why I think that evolution is the most important thing that has ever been discovered. Because if you look at Newtonian physics or Galileo or stuff like that, or all the things we talk about, evolution is the one theory that affects everything. It even affects literature and music and art. Because what do artists do? They're trying to bring out emotions so that you can connect with something and think about your history and where you are and meaning and purpose. Well, if you know when we got here, how we got here, where we came from, all of that informs the why. And the reason why evolution is attacked in some way by all religions is because if you think about it, it strikes at meaning and purpose and why we're here. And it rules out a lot of things that we have been taking for granted forever. So I would posit, as Daniel Dennett and a lot of other people, when he wrote Darwin's Dangerous Idea, is the reason why evolution is so attacked by so many people is that deep down they know that when you study evolution and you look at the fossil record and the history on this planet and the history in your own DNA with all these viruses and stuff, there is no planning. There are no goals. A rock comes out of space and wipes out the dinosaur and leaves niches for us, the mammals. We wouldn't be here without that rock. And the dinosaurs would have never been there without the Siberian traps, because before they got on, there were huge lava flows all over the Earth, which was the biggest extinction that has ever happened in this planet, allowed the dinosaurs to come in. So you look at the haphazard way of the way things are evolving and moving around this Earth. There's no planning, there's no goals, and some species make it, some people don't. 99.99% of all the species that ever existed are extinct. It's a sloppy history. There is no, from what we can tell from science, there's an ultimate goal behind that. That means that you're looking at a materialistic, deterministic world, and that is where the religions are going to attack, because that goes to the heart of their belief systems. And if you want to know about theistic evolution, which is the religious saying that, no, God used this wonderful process called evolution to create, I have a wonderful answer to that on my blog. I wrote about that. You can evoke that, and the Pope, the leader of 1.2 billion Christians, accepts evolution. Francis Collins accepts evolution. Graham Finley, who's a cancer researcher and a Christian evangelical, accepts evolution. Their form of evolution is called theistic evolution, or evolutionary creationism is the new label they're using. But their form is, I would posit, is not the way science has found it. And so there are some people like Nathan Lentz, who's a geneticist teaching at, I think it's John Day College. Anyway, he wrote a book on human errors and stuff. He's a great teacher. He is going to take the stand that let's just get them to theistic evolution and stop there, because then they'll leave science alone. Okay? I personally want to be a little bit more consistent in that. And I want to say, okay, does theistic evolution really solve the problem? Does it really accommodate theists? And I would say no. Here's another slide just to show you that they took a family of ERVs and they went through, and as you can see, there are 30 of them that are found on these various chromosomes that only are found in humans. There's another 14 that we have that chimps and bonobos don't have. Here's 6 of them that are shared by all 3 of these, plus the gorilla. Anyway, as you keep going back, if again, if disease happened at the fall and infections and all these problems at the fall, you wouldn't be able to do this. Phylogenetic trees, in my mind, are really solid evidence for evolution. And there are all kinds of phylogenetic trees. And I think, if you start to think about it, it's probably going to disprove their thesis of the fall. Okay, this is just a summary again. Random, they're nested. The trees match a whole bunch of other evolutionary trees produced by many different areas in science. And that this, to me, is, in my debates with the apologists, they're trying hard. And I have tried to research as best I can what they're saying. So they used to say that they're not random. They are. You know, they used to say that all kinds of things. And now the latest theory is they're trying to say something about VIGEs, which is something that they just made up. So we don't have time today. My site goes into, you know, if you're arguing with somebody and they throw something out, well, they're not random. You know, there are hotspots. And you want to know, I've got it in here. I think I've just addressed just about every creationist objection there is. And so my site should be a good resource for you. And because I feel that evolution is the most important discovery of mankind in the history of mankind, there are consequences. There are consequences about morality, about free will. I'm writing on my blogs monthly to try and get some of this stuff in. Very little of it is original to me. I've just done the research for you. And I have to address the objections and then the consensus by everyone. Let's see where we are here. Okay, I think I actually wrapped up a little bit early for questions. So that's the site. This is my anti-Christianity site also. And again, I don't have any theology degrees or anything, but boy, I found some really smart people. And if this DNA stuff if it means anything, you know, it doesn't connect with anybody, go watch my whale videos. They're much more fun. But this book just came out in paperback. It was really expensive as a hardback, and I wish I would have read this 10 years ago when I was doing all this stuff. But this is Graham Finley's book. I don't know if he's still practicing in the medical schools or not, but just the man is amazing. He's written well so people can understand it, and so I've taken a lot of stuff on there. There's also, like I say, other things on my site besides some of the consequences stuff in there. I just, you know, when you're debating with, you know, you're winning a debate with a creationist when they bring in abiogenesis. Does anybody know why? As soon as they start saying, well, explain how life started, you've already won.
One, abiogenesis isn't connected to evolution.
Exactly. Darwin.
Two, it's still an unknown. So.
Exactly. So Darwin wrote on the origin of species. He didn't write on the origin of life. Yeah, they're connected, but we can still know that humans got here by evolution, even if we don't know it starts. Anyway, so I have a blog on that. It's called How versus What, and they'll also throw that out. So a lot of contra-apologetics stuff here.
I'm just curious, what would you use to say if someone saying like a white person like, oh, whites and black, whites are superior to black because how would you use evolution to debunk that horrendous claim? So
the question was, how do you use evolution to explain why whites and blacks are not, superior in one or the other? And remember that Darwin was writing during the Victorian times in the mid-1980s, and he got a lot of things wrong. He didn't even know about genetics; it was totally unknown. So how you measure intelligence is very controversial, and so given the same opportunities, there are biological differences between men and women, and sometimes between races, but they're not so different that it means anything. So I think that when we have children, if you decide to have children, your body will make an exact copy of the DNA on the two sides and then mix them. But there's about 100 of those ATCGs that mutate. So even your children will have about 100 to 150 differences between you and them. And notice I didn't go into any kind of mechanisms like how this happened. How do you go from a chimpanzee or a gibbon or a gorilla to a human, for example? And that's a whole different story about mechanisms, natural selection, horizontal gene transfer, genetic drifts, blah, blah, blah. And so he's got a chapter in the back. I haven't gotten to it yet on new genes, how you make new genes. And so, because the creationists will say there's no transitional forms, even though there's a ton of them, no transitional fossils. They'll also say you can't make new generation. There's been they'll say there's no evidence for making new genes, but there is. You just have to go find where they've sought that. So basically, I would say that the different races that we see, are due to natural selection, fine-tuning different areas. But basically, we're all African underneath it. I mean, the whole story of how the world got colonized by humans is an incredible story that they've worked out through genetics mostly right now. So all of us outside of Africa came from a small group that left about 70,000 years ago. It's amazing what the DNA has shown. There were a couple of forays that went out at 120,000 years ago or whatever, but they didn't take. And so everybody outside Africa is from this one group that left Eastern Africa about 70,000 years ago. And now they're going into Africa and they're fine-tuning the groups that moved around and trying to figure out where exactly we came from. But at the DNA level, we're all Africans. Okay, you had a question back there?
Yes, I have a question. It's regarding a scientific word. So what's the word when two species, let's say a lion and tiger, are bred, but they're not able to reproduce? I was having this conversation last week, but I couldn't think of the actual scientific word when you breed.
Oh, that you get a hybrid that is infertile?
Yes, like infertile. I think there's another word for it where they're not.
Or sterile, infertile?
Yeah.
Yeah.
Because, like the saying, what I found interesting is how the polar bears are moving inland because of the Atlantic, disintegration or the
right.
snow. So now they're mating with grizzly bears to make new species of bear.
Well, they're probably so close aligned that that the only thing that was separating was geography. That the grizzlies didn't want to go up and live on the ice floes and et cetera. So, but probably if climate change forces them down in, they'll probably be, whatever. And I don't even know. I guess they are two different species. I'd have to go back and look. But at that time, are they really two different species? And that's where you get into taxonomy and there's splitters and lumpers in biology, and so the splitters will want to split everything out, and the lumpers will want to put them together, and sometimes they'll do a subspecies, when it has 3 names instead of 2.
I was going to ask, and this has nothing to do with that, but how do you compile some of your notes? Because where it's so big, what's some of the structure or format or note-taking site, or how do you kind of do it so you could have made the site?
I write it out in a Word or Pages, and then I've just been at this so long that when I run across a diagram or something, I go, that's exactly what I'm looking for. Then I'll pull it out and use it. And I have on occasion reached out to publishers and stuff to get copyright rights. And sometimes I get it back, and a lot of times they ignore me because I'm a little guy and I'm not making any money off this, so they don't care. If it's for educational, non-commercial use, they usually don't care. But I just read as much stuff as I can get, and I get to the point where I think I know the creationist arguments better than they do. And so if you're in the Midwest or the South, you're probably running into a lot of young Earth creationists. So ICR, AIG is Ken Ham, and there's another one in Australia called CMI. They're the ones that are producing most of the, when you Google, you get oh my gosh, there's all young Earth creationists. But there are other creationists out there, and so there's, by the way, if you look up here, there's a search engine on my blog thing here, and so it's really easy to find if there's a topic like. I've written a pretty good blog on intelligent design or unintelligent design, and there's stuff in there that you can't find anywhere else that I've run across. So, because a lot of the people you interact with may be interested in that. Well, what about intelligent design, or what about the multiverse? I have a thing on the multiverse, not because I'm a physicist, but because I found this amazing young woman, physics girl that Yeah, she's got a video on the Big Bang and raisin bread. Anyway, so it's you'll never look at raisin bread again the same way. It's only 10 minutes long. It's on my site. So I'm dropping in the best of the best. I used to be a teacher, and I'm trying to make complex things easily understandable. And especially when I run across stuff that I think is going to really hold up, because it's just been a long battle, my 40 years of going in and out with creationists. But this DNA stuff is holding up really good. And on my site, there's a thing about DNA scars. It turns out that the DNA breaks really easily. And we have mechanisms for repairing the DNA because it's an emergency repair. And Dr. Finley goes into this, and I just grabbed it from him. So the body, the host, will grab a piece of DNA from anywhere just to patch it in there, because it's like a bleeding artery, And so you can study the genome, and there are these patches. There's lots of them. And guess what? These patches are shared by different species. And you can build a phylogenetic tree using DNA patches, these emergency repair patches. It's the exact same principle that they're doing here. And so I wrote a blog on it. It's much shorter, easier to understand than the ERVs. And there's a phylogenetic trees you can make from the DNA repairs. It's just, this stuff is really holding up against the creationists. So.
Have you ever been to the museum in Kentucky, though, the creationist Noah's Ark thing?
I haven't been to it. I've watched other people go. That's enough for me. I was exposed to young Earth creationism when I was an evangelical for a couple years, and that's what got me out of religion, is because I went for a bachelor's degree in zoology, and I kept bumping up against what I was hearing in the pulpit versus what I was hearing there, and eventually the science for me was the door out. For most people, it's other things. It's the moral problems or the problems of evil or stuff like that, or things like that. But for me, it was science. And then about 15 years ago, when Bart Ehrman, James Taylor, and and also Christine Hayes at Yale started to put these lectures online about the Bible, it's like, oh my God, that was the second door. And so I've taken the best dirty stuff about the Bible, the things that they teach in the seminaries that you don't know about until recently with the internet, and that's on my site too. It's amazing. Yahweh was a pantheonic God under El. There's a reason why Elohim and stuff like that is there. It's just amazing, amazing theology. And so I've taken the things that I understand from them and Bart Ehrman. Like I said, not only are the Gospels anonymous, not only do they have contradictions, but there are 6 out of the 13 Pauline epistles are forgeries. The ending of Mark is a forgery. It has brackets around it. Pick up any Bible, and you'll find out that it's not there. That's the one that they use in West Virginia to do the snake dances and stuff like that. They're dying for something that's not even original. It's a forgery into the Bible. Anyway, really, really good stuff, and I've put that up there. But again, if you don't want to scare off somebody who's religious, there's the other site that is not. So anyway, thanks so much. The DNA stuff is, we don't need fossils anymore. We got DNA, and you carry it in your body. Okay.